And I'm recording the video for the people which is not here. Okay, welcome in the next lecture from biology. Today we would like to finish, or I would like to finish calculations and also start a pretty new topic, which is also related with the membrane potential. We are going to talk about action membrane potential and the membrane potential development. And next time we are going to continue with the muscle contraction or neuromuscular junction and related with that the muscle contraction. This is the plan for next time, so 26th of November. 3rd of December, we have to finish proteosynthesis. 10th of December, you will have a video, and also related to that, you will have some little questionnaire, some mini test, because I am not here. 10th of December, I have a conference. And before Christmas, we have the last lecture, 17th of December. Then we will focus on basics of genetics. And finally, the first week in January, I don't remember the date, but it doesn't matter. It will be a cell division, the last lecture from the course of biology. Also, I would like to show you your score. So here you can see that you have received the full number of points from the last calculations. So to get so Omar, he's not really coming to this course. I am not sure if he's still a student. Do you have some information about Ahmad Ziad Omar? Is he visiting some another courses with you?
He had COVID for 2 weeks, so he didn't visit, and he also has some problems with visa, so he's constantly visiting police, but we're not in contact with him.
Yeah, but I know COVID is a problem, but if it is not something serious, you can participate in online lecture. Okay, I will send him a message because there is not pretty much space to receive the credits if you are not coming to the course. I think the rest of you, 20 to 16 points, I think it's a good score. I'm pretty sure that if you just keep this working effort, you will receive the credits easily without a credit test or something like that. So just because of you are working continuously and you fill the questionnaires after each lecture, so that's a good point or good information for you. So that's nice. And now we are going to start or continue with calculations. Okay. So, okay, last time we had calculated the contributions of every cation or ion, every ion to the membrane potential. As you remember, the calculations number 1, the questionnaire and Google Forms, you calculated different values. So it was, okay, in the first example, it was minus 86, in the second one, minus 88, in the third one, minus 59, and in the number 4, 86, and last one, number 5, 56. So it's visible that the contribution of potassium in the resting state, it's always negative, something around minus 70, minus 80, minus 90. The contribution of I didn't write the numbers, the names of the ions, but it doesn't matter. So for chlorine, it's so also something around minus 60. For For sodium, it's usually a positive, something around plus 50, and for calcium, it's also a positive, something around plus 50 or even more. So this is just the values which more or less ranging for the resting membrane state. This value of the membrane potential is given from every distribution of every ion which is participated in this process. However, this is the ideal numbers. We neglect one very important parameter, and this very important parameter is the permeability for the membrane. This is the permeability of the membrane. So which can be, the permeability can be different for different numbers or different ions, sorry, for different ions. So this is the values which has been calculated for the 100% permeable membrane. However, it's not true normally. In the resting membrane state, for example, the membrane is not permeable for sodium, chlorine, and calcium practically at all. So the permeability ranges in level of units of percentage. For potassium, it's a little bit more than 90%. This is the values for the resting membrane state. However, in the action membrane potential, once the membrane potential is changed continuously from the negative values to the positive values, what is changed? The permeability of the plasma membrane, and therefore redistribution of ions. So let's look at that in detail. So how to calculate the real plasma membrane potential? It doesn't matter if it is a resting membrane potential, it doesn't matter if it is action membrane potential. The calculation is the same one. However, we have to use Nash-Bethes equation to calculate the ideal contribution of every ion, as we did before. And later on, we have to correct it because of the different permeability of plasma membrane for different ions. So let's consider a cell. I can recommend you just to follow my speech and just use just a simple paper and a pencil, and just to draw what I'm talking about. So this is a cell. So cell with plasma membrane. This is what is important now. The organelles inside, it's not important right now. For potassium, we know from previous lecture that potassium is in high concentration inside the cell and in low concentration outside the cell in the resting state, in equilibrium state. We are only talking about equilibrium state. So that's the reason that potassium would like to go outside the cell. It's captured inside the cell because of the negative charge coming from the organic molecules, as you remember before. It's opposite example for sodium. Sodium would like to go inside the cell because of its low concentration in the cell and high concentration outside the cell, and also because of the negative charge inside the cell. Chlorine would like to go inside the cell, again because of its concentration gradient. Its higher concentration is outside the cell. However, the electric force is repulsing chlorine because of the negative charge inside. For calcium, it's the same like for sodium. Calcium goes along the concentration gradient, so it would like to go to the cell because of its higher concentration outside the cell, and also it is attracting by the negative charge placed inside the cell. So last time we have calculated the potentials which will be given, or contributions to the potential given by every ion which participate on the membrane potential. So this value depends on the current concentration of every ion inside and outside the cell. So for example, we calculated these values. I just took the values from one example we calculated before. So it was calculated, it was done from Nash-Bethes equation. You understood perfectly because I check it with the questionnaire. So that's good point. We can continue. However, the permeability for sodium, potassium, chlorine, and calcium is not the same during the whole life of every cell. It's changed. And the permeability is a relative number. We can use the permeability in percentage as I use in our calculations because I think it's much simple for the imagination. So this is the permeability expressed in percentage. However, we can also use the permeability used as a relative, as a ratio. So it means that 100% it's 1. So 1 and 100% permeability means that the membrane is fully permeable for one ion, for example, potassium. Just to know that I am correct, you have some feedback. If you count together the relative permeability for every participating ion, it should be always 100% if you are calculating that in percentage, or 1 if you are calculating in a relative ratio of permeability. So how to calculate it? There are two main ways how to gain the result. The first one is using the Donan equilibrium. This approach arises again from the Nernst-Peters equation. However, it says that the equilibrium is equal on both sides. So it means that the sodium, potassium, or in this case, potassium and chlorine, they are compensating on both sides. So this establishment more or less follows the electrochemical distribution of ions, which is approaching the electrochemical equilibrium on both sides. This is just the definition which arises from the resting membrane state. We just put together, in this case, 3 ions. So potassium, sodium, and chlorine as the most important one is again the Nernst-Peters equation. However, it's much more complicated. We put together the concentration of each participating ion on both sides, extracellular, intracellular concentration, and also we multiply. Okay, we multiply by the permeability, which is different for different cations. Now I express the relative permeability not in percentage, but as a relative permeability. So 1 is 100%. However, based on the experience from last years, students have strong problems to calculate correctly this approach. Therefore, I found something similar, something easier way how to calculate the membrane potential if we include all contributions of ions which are participating on the membrane potential, and also without neglecting of the membrane permeability. So let's follow me. First of all, the aim is calculate again the membrane potential, which is given in this first simple example by just the contributions of 2 ions, sodium and potassium. So we have calculated last lecture that the contribution for some current concentrations of potassium is -92 millivolts. So this was calculated from Nernst-Peters equation. So Nernst-Peters equation, so R equal to RT ZF ln concentration in extracellular matrix, extra or out, and potassium inside intracellular. So the same was used for sodium. And now how to calculate the membrane potential? It's easy. We know that firstly, we expect that okay, again. First time we expect that the permeability for the plasma membrane is ideal. It's fully permeable, so 100%. However, indeed, in real case, it's not true. We have to correct it. So now we are going to focus on corrections. So yeah, it's in Czech. Why? Okay, so propustnost is permeability in Czech. Sorry. So individual contributions of ions will be like this. So I would consider that the permeability for potassium is 95%. For sodium, it's only 5%. So this is just the typical case of the resting membrane state. How to calculate the contribution of potassium to the total membrane potential? It's easy. Any ideas? How to calculate 95% from minus 92?
You multiply by 0.95.
Yeah, maybe it's a problem with microphone. I cannot hear you properly, Marek. Maybe try it again, please.
Can you hear me now?
Yes.
Yes. Okay. Multiply by 0.95.
Yes, it's good. So we have a point. Okay, so it's easy. You can calculate it like a percentage from some total value, or you can just do it step by step for people which are not so familiar. So you can say that 100% refers to minus 92 millivolts, and the question is which amount refers to 95%. So it's easy, as Marek said. The value which we were looking for is minus 87.4 millivolts. So this is the contribution of potassium to the membrane potential if we consider the permeability for potassium only 95%, not 100. So for sodium is the same one. So we will calculate 5% from 67. So by this way, for example, and it is 3.35 millivolts. So how to gain the final value of membrane potential? What about Eva?
The final value?
How we can gain the final value if we know the contributions?
You sum minus 87.4 with 3.35 millivolts.
Yes, very good, Eva. You have another point. So we just add together, make a summation of all contribution. So mathematically, membrane potential equal to x plus y, and it is like Eva said. So the membrane potential is minus 84.05 millivolts. So is it clear? Do you have some questions to this simple procedure, or maybe it's not simple, I don't know. Okay, try to calculate for your own. This is our values of potentials, the same one as previous case. So just calculate the value 95%. It was calculated before. So 1% from 67, the permeability is like this. The result 0.67 millivolts. Then we will add also chlorine. and its permeability is 2%. And also for calcium, and its permeability is 2%. So please try to do it for your own. I will wait maybe 1 or 2 minutes. Okay. So for chlorine, the value is minus 72 minus 0.72 millivolts. For calcium, 2% from 123 is CMN. Do you have a result?
Yeah, it's 2.46.
2.46, I think it's correct. Okay, and CMN. Okay. So, okay, Mariam, are you there? No,
Yes.
Mariam, okay, could you tell me the result of the membrane potential, the value?
Yes, sure. It's so it's 1.41. And yeah, and sorry, there's some. So it's minus 58.99.
Minus, yes, approximately like that. Okay.
Yes.
Okay, Mariam. Another point. Okay. Katarina, you are here also, yes?
Yes.
Okay, I will prepare a question for you. Okay. Now I also have prepared for you something like 5 examples, but before you will start to calculate it for your own, and then I will ask you what is the result. We are going to talk theoretically about action membrane potential. We introduced fully, I think, what is the resting membrane potential or equilibrium membrane potential. And now we have to explain what is the action membrane potential and what happened during the action membrane potential. So first of all, just remember that during just remember the distribution of the resting membrane state. Yeah. Katarina, could you tell me what is the distribution of all 4 ions in the resting membrane state? Just to repeat it. So where is potassium?
So potassium is inside of the cell because it has positive charge. The chlorine is outside of the cell. It has negative charge. The calcium has 2+, which is also outside the cell. And sodium is also outside of the cell.
Very good. Thank you, Katarina. So everyone who is now in the lecture has received just 1 point, so that's good. Okay, what happened during the action membrane potential? First, the channels for sodium are open, and sodium is really going inside the cell. However, it's not automatic process. We need the signal, the impulse, which is bigger or which is able to depolarize the membrane to minus 50 millivolts, from minus 70 to minus 50. So 20 millivolts difference from the negative to the positive values. Yeah, we are still in negative values, but we moved from minus 70 to minus 50 or minus 55. This is just the threshold. This is the threshold of the signal. You know probably the term which says all or nothing. This is exactly the value which refers to the summation of all signals coming to the cell. This is this threshold signal, this is just the initiation or just the information for sodium channels to become open. Yeah? It's a reaction for the change potential. These channels belong to the group of the voltage-gated channels. We will introduce them a little bit later, but just keep in mind that we have several types of channels. So this voltage-gated channel for sodium became open, and sodium is going pretty fast inside the cell. This process is responsible for another shift to the positive values of the membrane potential. We are getting to the plus 30, something around, and just the depolarization of the plasma membrane to the value around plus 30 is the signal for another channels, which are for potassium to become open. So in this potential, the sodium channels, voltage-gated sodium channels became inactivated. However, what is active is potassium, and potassium is going outside the cell. This is another contribution to the negative to the action potential. So how it looks like in the graph. Okay, this is this one I would like to see. Okay, first we are talking about the so this is the values of membrane potential. Okay, sorry. There twice this picture. I don't know. Okay. This is the plasma membrane potential in millivolts. So this is this X. So we are here in the resting state, something around -70. And this is the ion permeability, which is changed indeed for different ions, this one, permeability. So first, and this is time, just look at the unit of time, milliseconds. It's pretty fast. It's just millisecond, just 1 millisecond. This is the spike. This is the action potential. 1 millisecond and everything is down and back. So it's pretty fast. So first, we see the contribution to the action potential coming from the sodium ions. It's that one. Then I will change the color. Okay. And finally, this one is the contribution coming from potassium, which is going outside the cell. This line, maybe I will change the color again. It's the summation of all contribution. So the green one. So this is the summation, everything together. Yeah. If we look at the previous picture, but it was another picture. I don't know why I just put them in different order. We can describe the states of the membrane potential because the membrane potential is developing somehow, and it's pretty useful to remember the different phases of the action membrane potential. So number one, with the red color, it's the resting membrane state. As we calculated, introduce, explained many times before. Later on, we receive the signal. It's a spike. If the signal is strong enough, the sodium channels became open. So we are getting somewhere here, the sodium channel. So this is the threshold, yeah, all or nothing. So it's just the fluctuation. This curve is just the summation of everything. But here we see some fluctuation of the membrane potential. And once it exceeds or reach -50 or -55, the sodium channel just simply open. And the reaction of plasma membrane potential is enormous because sodium is really leaking or just going inside the cell, and the plasma membrane is changed to the extremely positive values. Once we get to the value around +30, adds another signal for potassium channels to become open. Sodium channels are now inactivated, and all the contributions to the depolarization of plasma membrane is coming from the potassium, which is just escaping from the cell. We are talking about depolarization or depolarizing phase. So depolarization. Now, once we reach the maximum of the spike of action membrane potential, we are talking about repolarization state, repolarization. So everything is returning to the resting state. Who is or what is responsible for this repolarization? It's the sodium-potassium pump. Sodium-potassium pump. Probably you know from biochemistry, it's the pump which consumes ATP to pump sodium outside the cell and potassium inside the cell. Because of the very intensive pumping of this equipment, of the cell equipment, we observe the decrease of the membrane potential back to negative values. So repolarization. However, because of the system is so active, sometimes we observe, or we often observe, something which is called undershoot. It means that the plasma membrane potential goes under the to the more negative values. However, in few milliseconds, this undershoot returns to the normal resting state. Questions?
What is the value of undershoot stage?
It's individual for different cells or different neurons, but typically it's around -55, -50 millivolts. It's a little bit more positive than the resting membrane potential. Just consider that the resting membrane potential typically for the majority of neurons, it's -50 millivolts. So the threshold is something which is more than 15 millivolts. Yeah. Is it clear?
Yes, thank you.
Okay, you're welcome. Okay, let's move to the calculations. Okay, here you have the contributions of potassium, sodium, chlorine, and calcium to the plasma membrane potential. And here down, there are I don't know why it is checked, but it's permeability. As I explained the same topic to my Czech students, the slides were in English, so some mistake happened in my presentations. So permeability is different. So we have 4 examples, and you can start to calculate the total membrane potential, which is given by the contributions of each participating ions. Is it clear?
Yes.
Yes.
Okay. So yeah, I give you something like 5 minutes. is ready, just say, I am ready. And I will just ask you for the results. And if all the results are correct, you will receive some points.
I'm ready.
Okay, Marek, you can tell me the results which you calculated.
The first one is minus 85.99. The second one, 39.62. And the third one, 47.75. Fourth one, 54.2. And the last one, 49.49.
Okay, let's see. Let's check. Okay. I think it was correct. Yes. Yes. Okay. Marek, you received 2 points because it was correct. Everything was correct and it was pretty fast. So that's fine. Okay, thank you very much. And now we can continue with another topics. So I also have prepared some calculations for you, 5 examples. I will send you the link by email. Okay, so let's continue with neurons. We are going to talk about the structure of neuron and about the action potential development, about the type of channels like leaking channels, voltage gated channels, chemical controlled channels. So that's the aim for today. So the neural system or neural net, it consists from 2 main parts. This is the central nervous system where belongs brain and spinal cord. The peripheral nervous system where are motor neurons and sensory neurons included. And the motor neurons can be distinguished as a somatic nervous system, which can be controlled by us. It's something which really is controlled by our, we are talking about the voluntary movements. And the second one, it's autonomic nervous system. This one is not really pretty much possible to control. We are talking about the involuntary responses. And we have 2 types of this autonomic nervous system. It's sympathicus, sympathetic division or sympathicus. It's simply the activating system, and this one is activated usually if we have some acute stress. So it dominates in the fight or flight reaction, as you probably know from the theory of stress. And on the other hand, parasympathicus, it's typically activated when we are in rest or if we are digesting. This is just a simple theory, which you probably know from physiology or related subjects. So what we can say about neurons? Neurons are cells, normal cells. However, they have something special. They have the antenna system, which is receiving information. We call this system dendrites. And also there is a cell body, which is normal cell body as we know. There is a nucleus, endoplasmatic reticulum, mitochondria, everything. And this big part, which is called axon. So dendrites as well as axon, these are the conductive parts of neuron. Dendrites are receiving signals and axons is conducting the signal. Here you can see it in more detail. You can see inside that there is really a nuclear cell, nucleus, endoplasmatic reticulum, ribosomes, Golgi, mitochondria, and so on. Everything which has to be in every cell, it's also in neurons. Here you can see that the red part, it's actually the conductive part. It's something like, there is a difference between the part which carries, or it's sensitive to changes of electricity. So we are talking about conductive part of the cell. So dendrites and some parts of axon. So do you know, so this blue part, it's a little bit different character of a tissue, or if it's not a tissue, it's a matter. It's a fat. Namely, it's Schwann cells. So these Schwann cells, they insulate axon, but not continuously, but some parts of axons. Periodically, it's switching the part which is insulated and part which is conductive. So it has a great reason because of it is provided like it is, because the signal, the transfer of the signal depends on the thickness of axon. Normally, if we haven't these Schwann cells, these insulating parts, the time of the action potential transfer or development, prolongation, propagation will be pretty long. You just imagine the distance from your brain to your thumb in your leg, in your feet. It's pretty long distance, and we have to speed it up because we cannot wait just to say, okay, my finger has to move. You have to move, just simply not to wait. And there are two strategies how to do it. We can make axons much wider, and because the diameter of axons or the speediness of the action potential development is increasing with the distance of the diameter of axons. So it means that if we would like to pretty fast reach the action potential from our brain to our feet, we need to have 1 millimeter and more wide axons. The practice, it's different. We don't have such wide axons. I think that the animal which has the widest axons ever, it's sepia. So this is the higher animal or the bigger animal which have this strategy or apply this strategy. For bigger or higher animals than sepia, they have these insulating parts of axons which helps to prolongate or propagate the action potential. Action potential, yes. It means that it's not necessary to continuously depolarize and repolarize, depolarize and repolarize the whole longness of axon along his whole body, but we can jump the potential just in these Ranvier knots, these red parts which are conductive. Is it clear? This part, or do you have questions? Okay, just remember Ranvier knot, Schwann cell, axon, dendrite, and also there is a synapses. Synapses, it's just the part which is responsible for the transfer of action potential. We have several types of synapses. Simply, it's the differing or the distinguishing of the synapses. There are two approaches. The first one is, or the first one depends if it is an electric synapses or if it is a chemical synapses. Here in this picture, you have the example of chemical synapses. Here you can see the synaptic cleft and also the vesicles where are some neurotransmitters and so on. And here we have the receptors. So that's the chemical type of synapse, and we have also electric type of synapse where the electrical signal is provided in more tiny system using the voltage gated channels. This is the first division, and the second one is based on the position of synapses. So here you can see the synapse which is just attracting the body of neurons, so we call this axosomatic. Also, it reached the dendrite, so it's another type. Also, we can find some synapses which are reaching the axon. Yeah. So there is a variety of synapses. It really depends how we use our brain. If we just provide there some pathways which are fixed, it could be a sequence of movement, or if we memorize something. By this way, the synapses are created. So here is a little conclusion. Maybe I can show you a video. Do we have a time? We don't have pretty much time. I will give you the video maybe in the end of the lecture, or I will post it to Facebook. So the conducting part includes the neurite or axon. This part is responsible for the receiving and creation of action potential and their propagation. Also, the part which exit or allows exiting of the action potential. This is just the endings of nerves, like neuromuscular junction, for example. This part is responsible for the secretion of neurotransmitter, and simply we call it synapses or synapse. Also, please remember that the response on the excitation of structures like nerves, sense, muscle cells, this excitation signal is always related with depolarization. So it means that action potential is the synonym for depolarization of the plasma membrane. So the switch to the positive values of the membrane potential. This process is called by impulse, spike, action potential, or response, all or nothing. And what is responsible for this process is the conducting part of the neuron. For sure, we have also, because we have to regulate this process, we have also some supressoric synaptic potentials. It's usually the parts of the membrane which are related with channels for anions, like for chlorine. We call this as a suppressive synapse or suppressive channels. We are going to mention them now, like later a little bit. And these work again the development of action potential. This is our break. This is the way how we can regulate the action potential development. So please remember that usually the membrane potential of neurons is around -70 millivolts. This is the equilibrium or resting state. So equilibrium membrane state. Please remember that potassium in this membrane state is pulled inside the cell because of the electrical force and pushed outside the cell because of the concentration gradient. The opposite is for sodium. Sodium is pulled inside the cell because of concentration gradient as well as because of its charge. Who is responsible, or what is responsible for the keeping the different concentration of sodium and potassium inside and outside the cell is the sodium-potassium pump. Also, there are two another ions which are helping to regulate plasma membrane potential, and their contributions are based on the changes of the membrane permeability. Now let's clarify a little bit the principle of the information transferred in the neural system. First of all, we have the, again in Czech, sorry. I don't know why. I have English slides in my Czech presentation. But I will translate it into English, sorry. So normally we have the resting membrane state. So resting membrane state, RMS. So the value of plasma membrane is minus 70 millivolts. So this is what we have in the majority of time. Secondly, what happens, some stimulus, the information come via this system. Maybe I will change the color. Okay, yellow one. So the information are coming through this dendritic system. And here we have the relay, the part of neuron which is responsible for the evaluation of the information which are coming. But it's not really in real state. We may receive only information via this dendrite or this one, not from all of them together. So it really depends on the summation of the space which is really provided in this part. Also, what is provided, the summation in time. So for example, this dendrite is stimulated in cascade of milliseconds. So they are coming, the pulses coming from one specific synapse. And therefore, this part provides the time summation of the signal. And depending on the intensity of the signals coming in time or in space to this part, we can observe the response, all or nothing. So it really depends on the intensity of the information. So just expect that the intensity of the information is enough to exceed the value minus 55 millivolts to open the voltage gated sodium channels. And now we are getting to the creation of action potential, which is firstly created here. And later it's developed along the axon to the end of synapses here. And now we are in synapse here, number 4, and it depends on the intensity again. If the signal would be transferred to another neuron or to the muscle or to some digestive cell, so to the target where the information should be delivered. Yeah. The synapses, there are again 2 types of synapses, the chemical synapses and electrical synapses. So let's focus on that. So chemical synapses, maybe this is much clearer for you because you probably learn about acetylcholine and glutamate synapses and so on. So how it works. So the action potential is developed along the axon to the end of chemical synapse. Because of the change of the plasma membrane potential from negative values to the positive values, it has a signal for these vesicles with acetylcholine, for example, or glutamate in neurotransmitter in general to be transferred to the end of chemical synapse and to be open. And therefore, the neurotransmitter is released to the synaptic cleft. So this space, synaptic cleft. By diffusion, the neurotransmitter is going to the opposite side, to the ending, or to the beginning of another neuron, or it could be a muscle, sarcoplasmatic reticulum, some part which is sensitive of every cell which is in the end of the cascade, for example. But what is important, there are channels which have receptors for such neurotransmitter. Neurotransmitter is bind to the receptor, and this is the signal for the channel to change the conformation and to become open. once the channel is open, it's usually the channels for sodium, potassium, and so on. Someone came. Okay. Okay, everyone is here in this lecture? Or my signal is too low. Could you hear me?
My internet crashed, so I had to rejoin.
Ah, it was you. Okay, not me. Okay, that's good information for me in this point. Okay, so let's continue to where I finished. So yeah, the channel became open and the ions can go through the channel. Yeah, there are several types of channels for sure. Channels for sodium, channels for potassium. So if just consider that these are for potassium or sodium, it doesn't matter. If the transmitter just reach the receptor, it's a signal for the channel to be open. And therefore sodium can go inside the cell. It's a cell. This part it's cell again. Just so this is a cell number one, and this one I will change the color. It's cell number 2. So is it clear that there are 2 different cells? Cell number 2. Yeah. So this is just the way how the information, how the action potential can be transferred from one cell to another cell. It doesn't matter if it is a neuron, motor neuron, sensoric neuron, or the second cell, it's a cell from the digesting system, it's another neuron. It could be also a muscle cell, whatever. In the second type of synapse, we call this electrical synapse. These are much tinier. So the way how they are connected, it's really the closer way how they are connected. So they have again, cell number 1, cell number 2. So the electric potential is just reached the end of the synapse, the end of the cell number 1. And this is the signal to open voltage-gated channels. So voltage-gated channels is these structures. They just became open because of they changed the conformation because of the change of the potential. So from -70 to plus something, plus 30, for example. So they became open. And once they became open, the ions can go inside and outside, depending which ion we have or which ion we consider. And the change of the concentration of the ions in this cleft influence the concentration of ions in extracellular space. And therefore, we change also the membrane potential of the second cell. And by this way, the voltage-gated channels, which belongs to the second cell, became open as well. Yeah, by this way, the action potential is transferred. So 2 ways how to do it. Is it clear?
Yes.
Great. I will show you one experiment which was done maybe 100 years ago. Yeah, maybe exactly 100 years ago. Yeah. Do you know Otto Levy? It doesn't matter. So first of all, just make a little conclusion. What is the chemically controlled channels and synaptic potentials related with that? What happened there? After the ligand is released and by diffusion reach the receptor, it creates a bond with the receptor. Neurotransmitter and the receptor creates a bond together. This is the signal for the conformation change of the channel. So maybe it's closed, so like this, and the change just allows to just slightly open the channel, and channel became open. channels are created from proteins, and it really depends from which amino acids these proteins are built up. So it also influences the charge of the proteins which are inside, or the amino acids which are a part of the or creating the proteins. So everything is about the charge, change of the charge. So let's look to Otto Lévy experiment. He did it, I think, 1921. Yeah, 1921. That's the date. He provided this famous experiment. Several years later, I think 1936, he received a Nobel Prize for this experiment. What he did? He prepared a frog heart. The first heart was prepared with nervus vagus, and he stimulated the ending of nervus vagus, and by the stimulation, because nervus vagus belongs to parasympathicus, so it decreased the heart beating activity. Yeah, by stimulation of nervus vagus, the heart beating activity of heart was decreased. He also had another heart, but this heart was without the nervus vagus. It was immersed in a liquid, in a physiological liquid, of course. And what he did, he just took a little bit of the liquid from the first heart, from the stimulated heart, and he put this liquid to the second heart. And what happened? The second heart again started to slow down its heart beating rate. So it was just the provement that we have some chemically controlled synapses or chemically controlled channels. So what happened on the molecular way in this experiment? By the stimulation of nervus vagus in the first heart, it was released to the physiological solution in the neurotransmitter. It was acetylcholine in this case. And because he took a little amount of the liquid from the first heart to the second one, he just took the neurotransmitter, acetylcholine, and put it to the second heart. And the reaction was the same one. The heart beating rate just slowed down. It was just the transfer of the action potential via the chemical way. So our membrane potential may react on the changes of the membrane potential, as well as on the presence or absence of some chemicals. Two ways of regulation. Is it clear? Questions? Okay. That's great. So we have indeed 3 types of channels. We already mentioned the chemically gated channels. So these are the receptors for neurotransmitters, as Otto Lévy proved. But here are also the voltage-gated channels, which react on the changes of the plasma membrane potential. I mentioned in the beginning of this lecture that we have sodium voltage-gated channels, which became open if we receive the threshold of the action potential of a spike, and this threshold is around minus 55 millivolts. We also have voltage-gated channels, which are for potassium, and they became open if the plasma membrane potential reaches the value 30 millivolts. But also we have another group of channels. We call this leaking channels or leak channels. It means that they are open practically without no limit, also during the resting state. And through these channels can leak, so it's not a stream. Leaking, if you have a leaking tap, so it means that once a second there is a drop of water. So little amount of sodium or potassium can go through this membrane. And what equilibrates the concentration of sodium potassium on both sides to keep the resting membrane state is sodium-potassium pump. So it's again the feedback process which may equalize the concentration of plasma of ions on both sides, or just to keep the desired concentration of ions or distribution of ions. So what we can say about chemically controlled channels, or chemically controlled channels. They are, so the channel is made of proteins. Proteins are made of amino acids. However, it really depends from which amino acids. Probably you will remember from biochemistry that we have several types of amino acids which creates proteins. We have 20 amino acids. However, some of them have different features. Some of them contain, some of them are negatively charged, some of them are positively charged. So the inner part, what is very important, is the inner part of the channel. If the inner part is created with amino acids with more or less negative charge, this channel is for cations. If the inner part of this channel is created by positive amino acids or positively charged amino acids, this channel serves for anions. Usually these chemically controlled channels are placed in postsynaptic member, so in the second cell, and maybe that's the type of chemically controlled. We have acetylcholine and glutamate channels. These two are the most important, and also chloride channels. These three types of channels, chemically controlled channels, we have. We have more than these three, but these three are the most important one. Acetylcholine and glutamate channels, this is the channels which supports excitation. They support the action potential development. On the other hand, chloride channels, they suppress the action potential development. So let's introduce the acetylcholine channel and its receptor. Acetylcholine, it's a neurotransmitter. We can find it in neural muscular junction, in vegetative ganglia, and in brain. This channel is permeable for sodium, potassium, and as well as for calcium. So let's see the picture. Here you can see that it's created from 5 units. Alpha unit, it's present there twice. The alpha unit has the sensitive part. We call this part a receptor, which is sensitive for acetylcholine. There are also another 3 units. It's globes, it's beta, gamma, and delta. However, the sensitive part, the receptor, is placed only on alpha unit. So you can imagine it like a tube, which is created from 5 meters, like 5 sticks. The tube created by 5 sticks, and normally it's closed. These 5 sticks are closed, they are together. If there is a stimulation coming from the acetylcholine, the conformation of the alpha unit is changed to create a little hole for the penetration, to be penetrated by ions. So you can see it like this. So here it's closed. The closure is created by the alpha units. Once acetylcholine just is bounded to this sensitive part, the conformation is changed and the channel is open. It's simple. The second channel which we are going to talk about is glutamate channel. Glutamate is a typical neurotransmitter in all vertebrates in brain. The channel is permeable for sodium, potassium, and for calcium as well. And these channels are related usually with the creation of memory, short-term as well as long-term memory. We distinguish several types of these channels or receptors depending on their on their ability to be stimulated by some artificial agent. So it means that just they provide some experimental just to use a cell, brain cell, where the channels. They use some artificial agent, and the channels which were stimulated substitutely instead of glutamate by some different channel, they put them into one group because the structure of this receptor may differ in different parts of the brain. So for example, I will show you one channel, which is glutamate channel, but it can be opened by N-methyl D-aspartate as well as with glutamate. So we are talking about the NMDA channel. This one is used for the transmission for calcium, and its activation is related with the long-term or permanent changes in the neuron system and creation of long-term memory. How it looks like, here we have a glutamate, very famous structure, and this is just this is the whole, practically the whole constitution of the synapse. Here we have the vesicles from where glutamate is released. Here is the receptor where glutamate is attracted, and for calcium the channel became open. Also, here is mentioned the cascade, which is related with the changes of our memory. It's these changes. Here you can see that there is required the assistance of more cells in our brain. So astrocytes, it's some supportive cells which are also helping to our brain to keep information. So this is the supporting apparatus of our brain. Also, we have to mention the anion channels. They are permeable only for chlorine. On the other hand, the anion channels, they have the stabilization effect to the equilibrium potential, to the resting membrane state. Because if these channels are open, the negative charge comes to the cell. So it's naturally the contribution of chlorines to the plasma membrane is negative, as we calculated many times before. So we are talking about suppressoric effect. There are two main neurotransmitters, ligands of these channels. The first one is glycine. The second one is gamma aminobutyrate, or sometimes famous like GABA. It's the abbreviation for gamma aminobutyric acid. It looks like that. Again, it's so again, this is a whole cascade about the suppression of the signal. But what is important that the channels for chlorine became open by the activation of the receptors of this channel. So I think maybe time is a little bit over because I would like to give you some time for the calculations. So please, I just finished in this slide and I am going to continue next time. I think I need something like 20 minutes to finish this topic, and also the next topic is also related with this topic. So it doesn't matter. We can finish it later on. So questions? So if you haven't questions, I'm going to send you the link for calculations. Again, you have 1 week to calculate it. Also, you can set to Omar that he can calculate it also the previous questionnaires. However, the deadline is Christmas. What happened? Okay. Okay, if you haven't questions, I wish you a nice weekend and just wait 2 minutes for the email.
Thank you.
Thank you. Bye-bye.
Thank you.
Bye-bye.
Thank you. Bye.
Bye.